Hi, my name is Gusum. I'm a student from NYU Abu Dhabi, and my major is biology.
Hi, I'm Dana, and I'm a dual degree student at NYU CAS and NYU Tandon, and I'm majoring in biology and CBE. This summer, we've been working under Dr. Kirsch on a project called The Role of a Ram-Mimetic Peptide in Cartilage Repair.
Walking, running, eating, driving and climbing stairs. You may not think about it, but you rely on your joints for all of these day-to-day activities. Now imagine what it'd be like to experience pain every time you do these things. This is a reality for 25 million people who visit their doctors due to chronic joint pain caused by damaged cartilage each year. Now you may ask, what is cartilage and why is it so important? Cartilage is a type of connective tissue and it is found all throughout the body. The three main types include elastic cartilage, hyaline cartilage, and fibrocartilage. The cartilage in your joints, also known as articular cartilage, is a type of hyaline cartilage. It is not as rigid as fibrocartilage but stiff enough to allow for joint support. It is also not as flexible as elastic cartilage, but still flexible enough to allow for easy and painless joint movement. Unfortunately, when articular cartilage is damaged, the repair process is often lengthy and painful. The first phase of repair after injury involves the activation of pro-inflammatory cells called M1 macrophages. After this, anti-inflammatory cells called M2 macrophages are activated. However, there is often prolonged activation of M1 macrophages, leading to joint inflammation and pain. Even worse, when the cartilage does eventually heal in an inflammatory environment, fibrocartilage typically forms instead of hyaline cartilage, causing the joints to be too stiff and painful.
To solve this issue, we must take a closer look at what's going on the cellular level. An important pathway that leads to inflammation involves two receptors called CD44 and RAM. After a cartilage injury, a long sugar molecule called hyaluronic acid, or HA, binds to these receptors. This signals the cell to transcribe the DNA of inflammatory cytokines into mRNA and then translate the mRNA into proteins. These proteins are then released from the cell to cause inflammation in the joint and ultimately cartilage damage. Our team at the Kirsch lab hypothesized that if we could stop HA from binding to RAM to begin with, we might be able to reduce the inflammation. To do this, we use a peptide that mimics the RAM receptor and binds to HA, thus preventing HA from binding to the actual RAM receptor and preventing the whole cascade that leads to the release of inflammatory cytokines. After treating the cells with these two RAM-mimetic peptides, we use three main techniques to quantify the reduced inflammation: immunofluorescence staining, enzyme-linked immunoabsorbent assay, and quantitative reverse transcription polymerase chain reaction.
Immunofluorescence staining involves using various fluorescent lights to identify certain markers in cells. This technique was used to determine that our cells were alive and to identify macrophages and to differentiate between M1 and M2 of macrophages. We also used an ELISA assay, which measures protein concentration using antibodies to measure the amount of CD44 which was cleaved or cut in cells. Previous studies have shown that CD44 is often cleaved when in an inflammatory environment. Therefore, we used an ELISA assay to determine the amount of CD44 cleaved in cells treated with the RAM-mimetic peptides as compared to untreated cells.
Reverse transcriptase polymerase chain reaction, or RT-qPCR. In this technique, the amount of mRNA of inflammatory markers is measured. To do this, mRNA is first turned into DNA using an enzyme called reverse transcriptase. Then, a polymerase chain reaction, or PCR, is used to make copies of the DNA. The faster the copies are made, the more mRNA there was in the initial cell, meaning that there were more inflammatory markers being expressed. Our results show that inflammatory markers decreased in cells treated with our RAM-mimetic peptides. This means that these peptides could potentially be used to treat joint pain caused by cartilage damage in the future and help millions of people.